my name is Vanessa Valencia, and I will be presenting on this paper called Pseudomonas aeruginosa, gshA mutant is defective in biofilm formation, swarming, and pyocyanin production. My partner is Michelle Patterson, and our papers both have to do with biofilm formation and virulence factors, but in two different bacterial species, where mine is in Pseudomonas aeruginosa and hers is in MRSA. So microbes are found everywhere. And so with the rise of antibiotic resistance, it is important for researchers to investigate these species such as Pseudomonas aeruginosa and see what genes are involved in these virulence factors, which vary from pyocyanin production, biofilm formation, motility, and antibiotic resistance. And all these factors can contribute to how sensitive they are to antibiotic treatment. So the aim or the goal of this paper was to investigate how a mutation in the gshA gene affects Pseudomonas aeruginosa. The main question was, how does the gshA transposon mutant of Pseudomonas aeruginosa lacking the gshA impact biofilm formation, antibiotic sensitivity, and motility of this bacteria? So I want to give some background on what Pseudomonas aeruginosa is exactly. So it is a rod-shaped gram-negative bacteria. It is an opportunistic pathogen, and it can be particularly problematic for patients that have cystic fibrosis. And this is because patients who have cystic fibrosis have this extra mucus in their lungs, which creates the perfect environment for this bacteria to replicate. And because it also forms biofilms, which are cells that are embedded in an extracellular matrix, it helps the bacteria adhere better to the cells, as well as be more resistant to antibiotic treatment. And this bacteria is also able to produce pyocyanin, which is a toxic compound, and this aids in its ability to be pathogenic, and it also helps it outcompete other bacteria that may be present in that same environment. So what exactly is glutathione? It is a low molecular weight thiol-containing tripeptide, and it is abbreviated gshA. So genes like the gshA and gshB genes encode for enzymes that are involved in glutathione biosynthesis. So that means mutations in these genes can affect virulence factors that include motility, pyocyanin production, biofilm formation, and antibiotic sensitivity. So the picture at the bottom is a image of the glutathione compound, and then the upper left picture is a pyocyanin producing plate for Pseudomonas, and you can see it's a greenish color, a little bit of blue, and then on the upper right, that is the compound for pyocyanin. For this experiment, they used the PA01 Pseudomonas aeruginosa strain, and there were 3 different strain types. So there was the wild type, which is the unmutated strain, and it was labeled MPA01. The gshA mutant transposon was labeled PW4072, and then there were 2 complement mutant strains, which were labeled TJB10 and BH01. All of the strains were grown in tryptic soy broth, also abbreviated TSB, M9 minimal medium, or the Luria Bertani broth, which is abbreviated LB. To assess the biofilm formation, crystal violet assay was used, and that would highlight the live cells, allowing the researchers to see the density of those biofilms. For the antibiotic sensitivity, they did this through serial diluting the antibiotics in the M9 media, and they measured the minimum inhibitory concentration, abbreviated MIC, and that was determined at the lowest concentration of antibiotics where there were no cells that grew after incubation. Motility was assessed by using specific swarming and swimming media plates. So now I'm going to talk about the data that was found through this experiment. So for this first figure, the A portion on top showed that in the M9 media, the gshA transposon mutant, which is this PW4072, was defective in biofilm formation when compared to the wild type, which is shown next to it in the MPPA01. And figure B on the bottom also showed defective biofilm formation for the mutant compared to the wild type. However, this portion was done in the TSB media. So for the second figure, for portion A on the left, it shows that the gshA mutant strains, and we have two in this case, which is PW4072 and PW9759, had decreased swarming ability when compared to the wild type. And for figure B to the right, they showed that the gshA mutant strains, once again PW4072 and PW9759, had decreased swimming motility compared to the wild type. They also did measure twitching motility, however, there was no difference found between the mutant strains that had the defect in the gshA gene versus the wild type. So the third figure shows that the gshA transposon mutants, PW4072 and PW9759, were deficient in pyocyanin production compared to the wild type. And this PW5404 strain that you see is a control strain. So this table shows that the gshA mutant had increased sensitivity to 3 out of the 7 antibiotics that they tested. And these 3 are highlighted by the blue stars, which was fosfomycin, kanamycin, and rifampin. So we can see that the MIC values for the PW4072, which is the mutant, for these 3 specific antibiotics is lower compared to the wild type. So we have 50 compared to over 100, 12.5 compared to 25, and 12.5 compared to 25 again. So through this experiment with the use of different plates and media, tests, and antibiotics that they decided to use, they found that the cells of Pseudomonas aeruginosa were stressed by nutritional restriction, which means that they weren't as functional in certain medias. They had a decreased biofilm formation, decreased swarming and swimming, decreased pyocyanin production, and increased antibiotic sensitivity when that gshA gene was mutated compared to the wild type. So in the discussion, the researchers talked about how the lack of the glutathione in Pseudomonas aeruginosa can have multiple consequences, and that the gshA gene is associated with different virulence factors. So when this gene is mutated, these phenotypes that help Pseudomonas aeruginosa become virulent are decreased, which decreases its ability to move, to establish itself with biofilms, pyocyanin production, and ultimately its ability to resist antibiotic treatment. So ultimately, the researchers were able to conclude that glutathione is critical for Pseudomonas aeruginosa to not only cause infection but to persist in an environment. And they also concluded that mutation of the gshA gene affects glutathione production, but it also impacts other virulence factors that are necessary for Pseudomonas aeruginosa to cause infection and persist as well.